All right, guys. So today we're starting Chapter 7, Life is Cellular. That is the title of the lesson for today, 7.1. So please follow along in your notes as we go through this. Highlight anything you need to, underline, circle, whatever works best for you. So what is the smallest part of any living cell that still counts as being alive? Any living thing. That is actually going to be our cells, the individual cell. So, what is cell theory? It consists of 3 points. One, all living things are made up of cells. Cells are the basic units of structure and function of living things, and new cells are produced from existing cells. So, we're going to revisit these 3 topics throughout this entire chapter. Now, let's talk about microscopes first. Now, microscopes like we have here in the science lab, these are relatively modern microscopes. The first microscopes weren't made until the mid-1600s, and you were starting to be able to see very, very small things. Now, an Englishman named Robert Hooke used an early microscope to look at a thin slice of cork, a plant material. This is the same cork material that you can find on a bulletin board. Under the microscope, it seemed like cork was made of thousands of tiny empty chambers that Hooke actually called cells. Because of this, we use the term cell in science today. Now we know that cells are not empty chambers, but they are openings that contain a huge array of different working parts. We call these parts organelles, and each of those organelles have their own function. We'll talk about those later in this chapter. So in Holland, a guy named Anton van Leeuwenhoek examined pond water and other small things, and he drew the organisms he saw in the mouth, and he called them bacteria. Now, one of the important things is you can see in the dotted line connecting C and D that these were actually moving. These are very, very small things that were moving. Now, these are not visible with the naked eye, so it was only possible to see these with the use of a microscope. Now, cell theory, Leeuwenhoek also saw that it was clear that cells were the basic units of life. They could move, they could divide, they could satisfy all those 8 requirements that we talked about back in Chapter 1. In 1838, German botanist named Matthias Schleiden determined that all plants are made of cells, and the next year, German biologist Theodor Schwann stated that all animals were made of cells. And then finally, in 1855, a German physician, Dr. Rudolf Virchow, concluded that new cells can only be produced from previously existing cells. But this idea has actually been around for about 50 years by a guy named Lorenz Oken, who was also a German. So these discoveries are summarized in the cell theory that we've already talked about. And remember these 3 things right here. All living things are made up of cells. Cells are the basic units of structure and function of living things, and new cells come from existing cells. So how do microscopes work? Most microscopes use lenses to magnify the image of an object by focusing light or electrons. We are going to get in the lab, and we're going to use microscopes to look at some cell samples, hopefully in the next couple of weeks. So a typical light microscope allows light to pass through a specimen and uses 2 lenses to form an image. You have what's called the eyepiece, and then you have the objective lens. So light waves are diffracted. This means they're scattered as they pass through. So light microscopes can produce clear images only to about 1,000 times. Now, that may seem like a lot, but we have to remember cells are incredibly, incredibly, incredibly small. Okay, so we can see individual cells, but we really struggle to see the individual organelles, and that's when different types of microscopes come in that we'll talk about in a second. So another problem with light microscopy is that most living cells are nearly transparent. So it's really hard to see them, and we actually have to stain them. Now, if you go to a hospital lab like the one I work in, we routinely stain cells all the time. And most of these stains are so specific that they will produce they will actually reveal only compounds or structures within the cell very specifically. So if we're trying to determine if you have a bacterial infection, we have stains that are specific to a very small number of bacteria. Some dyes will give off light when viewed under a black light. Fluorescent dyes can be used, and we can actually use these fluorescent dyes and watch how things move throughout organisms. So let's talk about electron microscopes. Now, electron microscopes can be used to see cells and cell structures as small as 1/1,000,000th of a meter. That's 1 nanometer. When we get smaller than that, we have to use what are called electron microscopes. So they use beams of electrons, not light, that are focused by magnetic fields. It's higher resolution, meaning it makes a higher quality picture, and it's also much closer. And there are two types. We have transmission and scanning electrons. So transmission electron microscopes make it possible to explore cell structures in large protein molecules. They can only pass through thin samples, so cells and tissues must be cut first into ultra-thin slices before they can be examined under a transmission electron microscope. It produces a flat two-dimensional image, and it can be viewed from there. In scanning electron microscopes, a pencil-like beam of electrons is scanned over the surface of a specimen. Because the image is of the surface, specimens viewed do not have to be cut into thin slices. They can produce three-dimensional images of the specimen's surface.